I suppose that we should maybe wait until a couple of minutes past, just to allow people to sort of sign in.
Not too many minutes, because we'll be up against the time.
Yeah, I was thinking that too. Okay, well, perhaps we'll just start. I've got a bit of an intro anyway in that anyway. Okay, so good afternoon, everyone. Welcome to the first Australian Cognitive Neuroscience Society Early Career Research webinar of 2022. My name is Ruben Rideau, and my co-host name is Isabella Bauer, sorry. I'm the ECR representative, and Bella's the student representative on the ACNS committee. Before we begin, I'd like to pay our deepest respects to Aboriginal and Torres Strait Islander peoples as the traditional custodians of the land in Australia, and recognise their continuing connection to the land, water and culture. We pay our respect to elders past, present and emerging. So these webinars are organised by the ECR subcommittee of ACNS and are aimed at supporting early career members within the society. We're going to be hosting another webinar later in the year, so keep an eye out for that. Also, I'd like to encourage everyone to come to the Australian Brain and Psychological Sciences meeting, which is a combined ACNS, EPC and OHBN conference that's being held in Brisbane in July, which looks set to host lots of interesting science. At the conference, there'll be 2 ECR events, which we'd really like to encourage everyone who's there to attend. On the first day of the conference, there'll be a workshop, and on the evening of the second day, there'll be a social event. I think both events are going to be lots of fun, so if you're attending the conference in person, then don't miss out. So our popular demand, today's webinar will focus on building skills related to using containerised environments, programming in MATLAB and R, and data visualisation. We've got 3 very good presentations, after which the speakers will take questions. If you have a question for one of the speakers, just write it in the comments and we'll go through them at the end. And also, if you miss out on any of the presentations, don't stress, because we'll be recording the webinar and we will make it available online afterwards. Our first speaker for the day is Dr. Angela Renton, who will be telling us about using containerised environments, and specifically how a new open source platform called Neurodesk makes this really simple. Angie completed her PhD at the University of Queensland and now holds 2 postdoc positions, one working for Professor Jason Mattingly at the Queensland Brain Institute, and another with Dr. Stefan Bollman in the Department of Engineering. She does really cool research using brain-computer interfaces, but it's the second role, working with Stefan on Neurodesk, that she'll be discussing today. I think that this initiative represents a really big step towards improving reproducibility in neuroscience, and I'm really confident that this way of analysing data is going to be increasingly common. So without any further ado, take it away, Angie.
Thanks so much, Ruben. Great intro. All right, I'm going to share my screen. Yep. We are play. All right. Hi, everyone. Thanks so much for tuning into this. This afternoon, I'm going to be chatting to you guys about the Neurodesk project. So for anyone who hasn't heard about Neurodesk yet, this is an open source and community-driven project aiming to help people to achieve open and reproducible neuroimaging data analysis using containerized workflows, which coincidentally happens to be the title of this talk. So I work with the Neurodesk Project as the sort of resident cognitive neuroscientist. And as a cognitive neuroscientist, my kind of toolbox of scientific software looks a little bit like this, which is to say that it's big and it's messy and it evolves from project to project. And a lot of the time, I'm using tools that are really kind of developed by a single developer or a small group of developers to a very specific purpose. And those developers don't often have the resources to support that software or those packages across lots of different operating systems or long term. And so having kind of scientific toolboxes that look like this really pose a big challenge for both accessibility and reproducibility in our development of analysis pipelines. There's a whole bunch of reasons for this, but I'll just go through a couple of them now. The first is that a lot of the tools that we use require Linux to run. This is especially the case for MRI data analysis, but also for some EEG and MEG tools. And for those Linux tools, they're often or most likely not available using standard packaging systems. And when that happens, we're asked to compile them from source. So if anyone who's ever had to do this before, if you're not a computer scientist, it can be an absolute nightmare. It never goes to plan and it takes ages. So even when we can install our tools, it's often the case that there are conflicting dependencies across different software, across different packages that we use. And even within the same tool, we can get conflicts between different versions. So if you're like me, you typically use a different kind of group of tools or a different group of packages or kind of a new version of each tool for every project that you run, which means that if you're trying to rerun something, an analysis pipeline that you had running like 4 years ago, it's pretty unlikely that you have the same versions of that software still installed on your computer because you're just not able to install multiple versions of the same software. On top of that, installing all of these tools on a single
I think we might have lost the sound from Angie.
Yeah, Angie. Wasn't sure if that was just me. She's also not progressing. Perhaps she's lost connection.
Yeah. Okay, well, bear with us. We'll quickly communicate with Angie and get back.
We could return to those if we can't find. Perhaps in the interest of time. Oh, she not? Perhaps in the interest of time, we should move on to Aron's talk, and then if Angie comes back in the interim, we can return to hers. Does that sound okay?
Sounds like a good plan.
Is that okay with you, Aron?
That's fine with me. Yeah.
Okay.
I'll try and share my screen here, and hopefully everyone can see that.
And I'll introduce you in the meantime. So we'll get back to Angie shortly. And now to introduce our second speaker, which is Dr. Aron Hill. He's a postdoc research fellow in the Cognitive Neuroscience Unit at Deakin University. His research uses a combination of brain stimulation and EEG to understand the neural mechanisms underlying brain plasticity and cognition. Today, Aron's going to be giving us an overview of MATLAB and R languages. When should you use which, the tips and tricks you kind of want to know when you're using these languages, and if you're learning from scratch, which should you choose? So thank you, Aron.
All right, thanks Bella for the introduction, and thanks for everyone to have me come and talk today.
So, sorry to interrupt, but I'm still seeing Angie's screen.
Oh, really?
Everyone else, yeah, I think you might need to dislodge Andy, like a host might need to, whatever, kick her out. Yeah.
Yeah, I should be able to do that.
All right. Cool. Awesome.
All good?
You can see my screen okay? Yes, great.
Yeah.
All right, I'll dive straight into it. Yeah, so today I'm going to talk about MATLAB and R. And the reason I chose these is because they're very commonly used in cognitive neuroscience, and they've got a big user base. I think that probably the most important thing I should say off the bat is I'm not sort of trying to pit these against each other at all. I think they're both very useful in their own way. And I think the reason I chose this as well is because they often complement each other really well. So rather than being at sort of a MATLAB versus R situation, I kind of see this as using both. And the reason for this is that they've got a large user base, like I said, they're regularly updated, and they've got a lot of cross-platform support as well between Mac and Windows, for instance, which makes them very, very usable. So what I'm going to do for this talk is basically just go through some features that I found particularly useful when I was learning to code and that I still find useful now. This is obviously going to be a very sort of macro level approach, just given the time. And also it's sort of pitched largely at people who might just be wanting to start to get familiar with these programming languages. So I apologize to people who are already quite fluent in these languages that will find this probably, they'll already know these details. The first one seems pretty obvious, but I think it's important and a lot of people don't do it. And that's just relating to actually customizing your environment when you start using either MATLAB or R. When you first download these programs, you're greeted with these, fairly bleak environments. They're sort of pretty white and pretty bland. And often I find they're a real strain on your eyes. So the first thing that I always recommend doing is working out a situation that works best for you. So this is, for instance, my MATLAB environment here. You can adjust this easily using the home layout function on MATLAB. Similarly, if you're using R, you can go Tools, Global Options, Pane Layout, and this allows you to sort of customize the environment to your specific workflow, which I find is really useful to do. The other thing as well, as I mentioned, is sort of just around changing your color scheme. Again, I find the white screen when you're looking at code for hours can be really hard on your eyes. There's a couple of ways to do this. In MATLAB, you can do it through the MATLAB preferences, but I've found this pretty clunky. So one thing that I've found useful is actually there's some custom-made toolboxes in MathWorks. If you go to their file exchange, you can actually, one I use is called MATLAB Schema. You can download this for free, and basically it will allow you to just run a line of code and change through different schemes that you can then load into your MATLAB to make it much nicer to look at. You can also do the same thing in R. R's a little bit more, it's got a lot more built-in options, I find. So you don't actually need third-party software. You can literally sort of just go Tools, Global Options, and it will already have a list of different themes available for you to choose. The second thing I thought I'd touch on, which again, I guess is pretty basic, but I find a lot of students and people starting out still seem to run into trouble with this, is installing toolboxes. This is particularly for MATLAB, where it's not always overly intuitive. And the thing with MATLAB is some toolboxes come as add-ons. So they're actually proprietary toolboxes that are developed by the MATLAB software developers. And they need to be installed when you install MATLAB, typically with the MATLAB installer. And exactly what's available depends on your university license. But a few that I would highly recommend for EEG analysis are things like the Signal Processing Toolbox, Statistics Toolbox, and the Image Processing Toolbox. Particularly the Signal Processing Toolbox for EEG, because it contains a lot of filters and power spectra functions and things that are often used. And one thing that I often see happen as an error when people are starting out is they might not have one of these toolboxes installed, and then they'll try and use someone else's code, or perhaps run third-party software like EEG Lab or Field Trip, and it will error because it's trying to call functions that just simply don't exist. So a great feature in MATLAB is in the command window, you can just type VER for version, and it will actually give you not only all of the detailed information about the various toolboxes that you have installed, but also about the MATLAB version itself, which can be really useful, for instance, if you're wanting to install new software and you want to know if it's going to be compatible. Similarly, on R, you can also directly do this through the Packages tab, which will then give you a list of all of the different packages that you have installed. Just another note on installing toolboxes. I mentioned that the MathWorks ones actually, you can select when you're installing MATLAB, but third-party toolboxes, so things like EEG Lab, Field Trip, or anything else, need to actually be installed manually. And what I found really helpful is just finding a way to keep track of those, because as you sort of progress, you end up sort of collecting more and more of these toolboxes, and it can be easy to lose track of them. So what I always recommend people do when they're starting is MATLAB will create a folder, typically in your documents folder. What I always do is just create a new, say, external toolboxes folder or something like that. And then every time I download a new toolbox, put it in there. And that makes it really easy to keep track of the toolboxes that you have. And in addition, also makes it really easy for backing up if you need to, update your computer for some reason. The other thing that's really important as well with MATLAB, is making sure that you always set the path to any toolboxes you use so MATLAB can find it, which again, I often find people starting out often run into issues with this, with MATLAB simply not finding toolboxes that have been downloaded. With EEG Lab as well, just touching on this very briefly, there's a nice way to do this through the Manage EEG Lab extensions, where you can literally do this and then select any of the toolboxes there. And the great thing about doing this is it will directly add them to your plugins folder, so they're ready to use every time you load EEG Lab. For R, I think it's a lot more intuitive in this regard. R Studio at least directly communicates with the CRAN repository, which holds all of its packages that it uses. So it's great for installing packages, because you can simply type install packages and then the package name in R, and it will automatically download it from CRAN. And then similar to MATLAB, where you would set it to path, you can just simply type library and then the name of the package, and that will load the package ready to use, which is great. So I thought what I'd do now is just talk very briefly about sort of building a script. And again, this is obviously based on people just getting started in coding. The example is in MATLAB, but I think this is, transferable to most languages. I think the biggest thing that people starting out coding face is writing scripts from scratch can be incredibly daunting. You've sort of just got this blank page and you're expected to just start writing code. So what I found really helpful, and what I still find helpful, is actually first outlining sections of a script without using any code, just using comments, and then basically going through and filling in the gaps, because it allows you to break down the analyses into really achievable chunks or to steps, and then to sort of go through those systematically, which can be really helpful. So to give a really quick example, I mean, this is an example of some MATLAB code for just loading EEG files, or TMS EEG files in this case, and then pre-processing them. So what I like to start off with is something like this. Again, no code here, just comments, just an overview of what the script is up the top, and then break down all of the different sections that I want to achieve with that code. And then instantly you already don't have that blank page. So it's not as daunting, and you can sort of then sequentially start to add code as you go along. I apologize, this is a bit of a busy slide, but essentially what I would do next then, and something I'd highly recommend doing, is at the start always having a heading and a broad description, and then even maybe a more thorough description underneath for your code. This isn't something I originally did, but I do it all the time now, because I think it's so useful when you share code, if someone else can get it and instantly look at the top and see exactly what the code's going to do. And it's also great for if you're like me and you sort of write code and then you come back 6 months or 12 months later and you have no idea what you've written. Instead of having to go through all the code and decipher what it's done, you can just look straight at the top and already go, oh, okay, that's what this script was written for. A couple of other things I found really helpful, again, around sort of particularly sharing code, is consider using functions like the file separator or file sep function. That can be really useful because, again, MATLAB and R are, cross-platform. So you can, sometimes you'll be sharing code. I work on a Mac, sometimes I'll be sharing it with people who use a PC, for instance. And there's issues with forward slashes and backslashes and incompatibility between the operating systems. So something like as simple as using a file separator function in place of a forward slash or a backslash can be really helpful, and means that code's likely to run much more seamlessly when you share it, which can be useful too. Another thing I found really useful, particularly on a Mac as well, is you're often having to get path names for your data. What you can do is you can just simply right click on a folder and then hold down Option, and it will allow you to copy whatever folder or path that you're in as a path name that allows you to simply paste it into your code, which makes things a lot easier and a lot quicker. Sort of related to this as well are shortcuts. I mean, obviously in writing code, you tend to use a lot of shortcuts because it just speeds everything up and makes it a lot easier to run. One of the frustrations that I've found going between different platforms, say MATLAB and R, is that often they have different shortcuts for performing the same task. And this can be really tricky because you'll be doing something in MATLAB, then you'll switch to R and you'll just keep mixing up the shortcuts. And so one of the things I found incredibly helpful is actually to consider customizing the shortcuts within either R or MATLAB to make them consistent across the platforms. So in MATLAB, for instance, you can go preferences, keyboard shortcuts. And then what you can do is for typical ones you use all the time, like for instance, running code, running a section of code, you can just make them the same. So when you switch between the two, you don't have to worry about remembering different shortcuts for different software. Finally, just a quick note on the, this is for EEG Lab specifically, which is a MATLAB package for EEG analysis. A really great feature of this is the EEG-H function, which I'm sure people who use EEG Lab are familiar with, but for anyone starting out, the great thing about EEG Lab is, in addition to code, it also has a really intuitive graphic user interface. And one of the things you can do is you can actually do anything on the graphic user interface. And then say, for instance, in this example, I wanted to re-reference my EEG data, for instance, to the average reference, which is commonly used. And then if I want the code for that, it has this great feature where you can simply type EEG-H in the command window, and EEG Lab will kindly provide you all of the code that it used to run that graphic user interface function, which is fantastic when you're starting out building scripts and working on scripts, because you can work out how to do things and then turn them into code, which is really, really useful. All right, so just a few, I guess, final tips that I've put here that I found really helpful, again, when sort of starting out with these platforms. I think it's a really good idea to sort of find others in your lab or in the wider university who are at a similar stage to you, and to sort of just try and meet up and sort of regularly discuss code and various tips and tricks that they might have. And I find that can be really, really useful. Another thing as well is just practice adapting other people's code. If you've got colleagues that have been working on certain code that's maybe similar to what you're trying to achieve, it can be great to grab that code and just try and play around with it to try and get it to do what you want it to do. It can be a lot less daunting than starting from scratch, and it's a great way to sort of go through different functions and work out how they work, or different sections of a pipeline. Another thing would be just taking opportunities wherever possible to work with collaborators who are really more experienced than you in coding. I got an opportunity to do that during a postdoc, and it was just fantastic. I just learned so much just working constantly around other people who were much better than me at coding. Also, if you can find a mentor who's willing to teach you, that's also fantastic. And the final thing, and I think really important thing, is just to dedicate time to learning how to code, because it does take time. And I think often these sorts of things can be easily sort of placed on the back burner when we have more pressing things. But I think if you can sort of schedule in time where you're just going to sit down and learn to, spend some time learning coding, it can be really, really useful and pay off in the future. All right, so just I'll pretty much finish up here. Just in the interest of time, just a few additional resources. Really useful things I've found for starting out is Mike X. Cohen, who's an EEG researcher who does a lot of work in MATLAB, has some great books and courses online, and a couple here that I've got. MATLAB for Brain and Cognitive Scientists, which is really great, and also Analyzing Neural Time Series Data, I'd highly recommend. A bit of a shameless plug for a pipeline that I was involved with, if anyone's interested in automated EEG pre-processing. Also some other links here as well. I'm happy to share these slides as well with anyone who's interested if they want to get access to the links. But I think I'll leave it there in the interest of time. So thanks a lot.
Oh, Ruben, you're on mute.
Classic. Sorry, I said thanks, Aron. That was a really amazing talk. I feel like I've been programming for 10 years, and there were things in there that I've only adopted, recently, and I wish I had known when I was doing my PhD. So yeah, really, really useful. So we've got Angela back now. We'll take questions at the end, but we'll just keep going with Angela's talk, and then we'll get on to Andy's. So Angie, are you there?
I am. Sorry about that. The irony of having technical difficulties while delivering a technical
I'm glad that we didn't
workshop
break from tradition and we had some technical difficulties.
I also can't start my video again because I think you need me to make to be host.
Yeah,
but let's do that. it's also okay to deliver it without the video. We've got slides.
What happens when you try to share?
The host can share, only the host can share in this meeting. So I think I need you to make me host.
Oh, okay. Hopefully I have that. Oh yeah. Wait. Let's make it so first. Okay, try now.
All right. I'm back. I really am so sorry about that. It's never happened to me before, and I'm really hoping that it doesn't happen again because I've not done anything differently. I'm really not sure what went on there. All right. Can someone with a mic on just tell me if they can see the screen so that you still have me?
Yes.
Awesome. Okay. I'll just go back a bit. I believe you guys lost me around about here. I was giving a talk to myself for a little while there. So yeah, I was just talking about the difficulties of compiling from source, and specifically all of the challenges for accessibility and reproducibility that scientific software poses for our data analysis pipelines. So yeah, one of these difficulties is that we often have conflicting dependencies across different tools. So even when you can install tools, a lot of the time you can't get two different scientific tools working at the same time because they require different things in order to work. And maybe something that's more familiar with people is this idea of conflicting tool versions. So if you have a specific bit of software, usually you can only, a lot of the time you can only have that version installed at the same time. A good example of this is actually GLab, which Aron was chatting about just before, in that you can only have one version of the GLab in your path on MATLAB at any one time. So if you want to, in a new project, which is something I often do, upgrade to something new with sort of new functions, if you want to roll back to a project that you had running 3 or 4 years ago, that probably worked with a completely different version to what you have now, and you're going to have to scale back your whole environment to try to run that that code or that analysis pipeline again. On top of that, there's a lot of issues with mobility in this sort of idea of having to have everything installed on your workstation, because it is so difficult and so time consuming often to install all of our different software. That means that when we want to move to a new computer, potentially to collaborate with someone new or to sorry, I'm just double checking that I'm still sharing, or to upgrade to say a computer with more resources, like a high performance computing cluster, we have to spend a lot of time reinstalling all of that software and trying to replicate our environment. And it's often not possible to do that with the same versions of software, especially if we're moving across operating systems. For example, from a Windows machine to a Linux machine, we probably can't get exactly the same software versions either way. And that means that we can get different results between our analysis pipelines. And a lot of the time they actually completely break because we don't have access to exactly the same functions. Now, this isn't even just the case between different versions of software. There is this paper that was published in 2015 by Gletard et al., where they had basically, they try to run a bunch of fMRI analyses using the exact same data and the exact same software versions across 3 different versions of Linux. And they found that the results of these analysis pipelines actually differed significantly depending on what version of Linux they were using. Now, this is a huge problem this should be a huge problem for us as scientists. Reproducibility is basically one of the core tenets of the scientific principles. And if we can't even replicate our results using the same analysis pipeline and our original data, first of all, we've not even really shown anything, but we're also massively hindering kind of scientific progress, because that means that other people can't kind of take our original analysis pipelines and build on those to create something new. Now, thankfully, there is something that we can do about this. And the solution comes in the form of something called a software container. Now, I've never heard of a software container before about a year or so ago. So for other people that are in the same boat, I thought I'd start with kind of a brief primer on what these are. The basic principle is that when we develop a piece of software and install it in a computer, it's not really a host unto itself. It's actually dependent on a lot of other things. There are system files and there are other packages and programs on your computer that it needs to run. So when we try to move the software to another computer or sort of change up other things in our computer, this can often break the program because it doesn't have those dependencies that it's reliant upon to run. And this is where software containers come in. So these are basically packages that include not just the software, but also every single dependency from low-level system files up to sort of other programs or photos or icons on the computer that it might need to run. And we package it all up together in a container, which can actually run on any operating system in any computing environment, and it'll run the exact same way no matter where you put it, because it has all of the dependencies and it has access to the exact same dependencies no matter where it is. And what's great about this is that it also contains all of these dependencies along with your software, so that they're kind of isolated from the rest of your computing environment, which means that you can have different versions of the same software installed in parallel in different software containers. And you can also have packages with potentially conflicting dependencies. So what software containers give us is standardization, portability, reliability, and reproducibility. Now, I should mention that I got a lot of these figures from a great YouTube video put together by the Australian Research Data Commons on software containers. So if you want to know a little more, I highly recommend watching that. And you can find the link to that on our Neurodesk website if you Google it, which brings me to the Neurodesk platform. So Neurodesk was built by a group of very smart people who work with neuroimaging data and recognized all of these problems and also were familiar with software containers and thought this is really something great that we need to bring to other cognitive neuroscientists so that we can make these reproducible data analysis solutions available to everyone. And so basically at the core of the Neurodesk platform is this huge library of containerized neuroimaging software. There's over 200 gigabytes of software spanning from structural and functional fMRI analysis, MEG, EEG, physiological data, you name it, they have the solutions, have packages that will analyze it. And it's an ever-growing library. Now, what's really, really fantastic about the way this is built, what's really fantastic about the way this is built is that it's actually, all of the software is hosted, all these software containers are hosted in a cloud. And in order for you to access them, you never have to install anything locally. So they've built this accessibility layer that allows you to run software virtually as though it were installed locally on your computer without ever having to install anything. So there's two main different ways that you can access the software. The first is through a platform called Neurodesktop, which is a virtual desktop environment. Now, it's really important to know that this is very different to a virtual machine. So if anyone is familiar with a virtual machine, that is a kind of mini computer that runs within your computer that has a whole operating system and is really kind of memory intensive and corners off some of your system resources. But allows you to access a different operating system. What Neurodesktop is a containerized environment containing software that launches really quickly. It only takes a gigabyte to store, and it runs as though you are, inside of a desktop environment. So you can launch all of these different neuroimaging software and packages just as though they're applications installed on your computer. The second way of accessing Neurodesk is through something called Neuro Command. So Neuro Command, it allows you to access the same software, but launching programs through command prompt. So this is something that you can use if you are using high performance computing clusters. And what's really exciting to me is that it will also work in Jupyter notebooks, which means that you can embed the software from Neurodesk inside of, say, a Google Colab notebook. Where that's relevant is if potentially you're writing up a manuscript, what you can do is include a link to a Google Colab notebook that runs all of the key analyses and reproduces all of your key findings just at the push of a button without anyone ever having to download any data or install any software locally. So these are our two sort of ways of interfacing with Neurodesk. We've got Neurodesktop and Neuro Command, and together this allows you to run an analysis across virtually any computing environment that you can think of. And across all of these different computing environments, you have access to exactly the same, computing environment. That means that your scripts and your analysis pipelines will run exactly the same way, no matter what computer you're running them on, reliably and reproducibly without kind of an issue of running into version differences or differences in the way, things are computed. So really at the core of the Neurodesk platform is the community of cognitive neuroscientists that are using this tool. They're the ones that are interacting with the software through Neurodesk. But what the developers at Neurodesk have done is they've also kind of democratized the tool so that it's really easy for anyone really to contribute new tools to Neurodesk. So there is on our GitHub repository a tool that makes use of GitHub's continuous integration so that anyone from the community can upload a new recipe for a new container containing a bit of software. And I think it's at midnight every night, there's a script that runs and will build this into a container that will be integrated and deployed across Neurodesk for anyone globally to use. So almost immediately the whole world can have access to that new tool that you contributed to Neurodesk. So if you are listening to this and thinking this is something that you really want to try out yourself, I highly recommend that you head to the Neurodesk website, which has a ton of documentation for how to get started no matter what operating system you're on, in terms of using all of our different tools. So I've already described Neurodesktop and Neuro Command, which is our desktop and our command line interface for launching different containerized software. You also have access to the kind of library of containers that are available on Neurodesk if you just want to either download a single software container or you want to contribute your own recipe for a new bit of containerized software. Coming very soon is something that will be available worldwide called Neurodesk Play, which is a completely virtual version of Neurodesktop so that you don't have to install anything at all locally and can just launch a version of the desktop in the web. But thankfully, all of us here are in Australia and New Zealand, so we already have access through the Nectar desktop service. So this is a free cloud-based computing platform that's available to all Australian and New Zealand researchers. And if you just go to this link here, you will be able to access Neurodesk literally just using your institutional login. You follow the link and log in, and you'll have access to a desktop environment with, I think it's 8 gigabytes of RAM and 16 cores. And yeah, that entire 200 gigabyte array of software for neuroimaging data analysis. And this really will work anywhere. I tested this out just before. I'm on my iPad right now and I can launch that desktop environment. So I could be analyzing my fMRI data from a cafe somewhere on my iPad, which is pretty fantastic. And the final thing on here is a tutorial for how to get started with Google Colab if you want to make one of those Jupyter notebooks that will run your analysis wherever you are. There's a ton of other stuff on this website. One page I highly recommend you check out is this tutorials page, where basically there's a bunch of tutorials for how to get started with tons of different software that we have included.
Sorry,
in
Angie,
Neurodesk.
just in the interest of time, if you could just try to wrap it up quickly.
Oh,
just so
absolutely, that was the end. So I just wanted, that's all good. Sorry, I didn't see my 2-minute notice. Basically, I just wanted to conclude by thanking all of our collaborators and granting bodies around the world. You can see there's a lot of people involved in making Neurodesk happen.
Thanks. Great talk, Angie. I genuinely think this is the way of the future. Our final, sorry for cutting off. Our final speaker is Associate Professor Andrew Purforce, who will be sharing with us the do's and don'ts of data visualization. Andy is the Deputy Director of the Complex Human Data Hub at the University of Melbourne School of Psychological Sciences, where he runs the Computational Cognitive Sciences Lab. As someone who often skips straight to the figures when reading papers, I think that data visualization is a really important topic that as researchers, we seldom receive any training for. And like it or not, I think that data visualization can have a significant influence on the impact and reach of scientific results. So I'm really thankful that Andy's here and speaking with us today on the topic.
Great, thank you. I'm going to share my screen and I'm glad you think that about data visualization, because that's what I think as well. This is in fact going to be like basically 15 minutes of thinly veiled ranting about why visualization is good and what makes it good. I just sort of wanted to start with, kind of open question for everyone to think about for a bit. When you think of data visualization, what do you think of? And I think, one of the first things that many people think of is that it's essentially about coding, right? It's technical skill to be able to create figures in whatever your software is. And it is partly that. I mean, it's not inappropriate that I'm giving this in a technical webinar, but I would say that's actually the least interesting and important aspect of data visualization. And often if I push someone further and say, okay, it's not really coding, what is it? Then there's sort of, well, maybe it's about aesthetics, right? So what makes something a good visual is that it's pretty, it's nice, it's pleasing. And again, partly, but for science, what makes something pleasing and aesthetic usually is because it's communication. So what we're doing with a good figure, and what we should always be aiming to do with a good figure, is we're telling a story. I mean, that's what we're trying to do in a paper as well, right? And so our figures should be structured to basically communicate a message. And so I think that is much more getting at the core usage of the figure. And that is in fact why, say, people like Ruben and probably many others, including myself, when we read a paper, one of the first things we do is look at the figures, right? Because the core of the message is often there. So I think we're starting to get more at the core of what data visualization is for. But I would actually argue that more than any of those things, data visualization is really a scientific tool for discovery. And this is not often realized or taught, partly because data visualization isn't often realized or taught, but also because it's often taught backwards as a thing that comes at the end. And I strongly believe that that's not what it is at all. It's a tool for communication, but the first person we're communicating to is ourselves, basically. We want to figure out how to take this lump of data and get insight from it. And figures are much more powerful than statistics for that. And so just to sort of, I think many of us who've been here doing it long enough kind of realize it, but I really want to drive this home with a couple of examples. You might have seen some of these examples before. The first one dates back like 200 years, back when they were worried about a different pandemic. I guess cholera wasn't a pandemic, a different disease. They had the novel idea of, hey, let's get some data on this cholera outbreak. And the data looks something like this, right? They got people, their age, lots and lots of variables. At this point, people didn't know about the germ theory of disease. They didn't know what might cause cholera. And this guy, John Snow, had the brilliant idea of let's visualize it, right? And so what he did is he visualized a map.
Sorry, Andy.
Yeah.
I just can't, I don't think your slides are progressing.
Are they not?
We can only see the first one.
Seriously? All right, hang on a second. Let me try that again.
We were visualizing what you were saying.
That's
okay.
Right, yeah, all right, okay. Okay, hang on a second. Let me try that again. All right, is that working? Do you see those?
Yep, I can see this.
All right, okay, I'm gonna try pressing play now and see is it progressing?
Yeah,
It is,
that's
all right.
Awesome.
Okay, so yeah, imagine there were a lot of cool, there were so cool pictures before. All right, anyways. So what he did was he had the brilliant idea of visualizing. And in particular, what he did was map, here's the town, London, where it occurred, and map the cholera cases. And, as soon as you visualize something, something pops out, which is, hey, they seem centered somewhere. And then he had the additional brilliant idea of visualizing, hey, let's think about where the water pumps are, right? And again, this is before the germ theory of disease. There's no mechanism under here. But as soon as he did that, it's like, I have an idea of what's going on. And lo and behold, we removed the handle from this pump. This pump was, had water that was infested with cholera. As soon as you removed it, the cholera outbreak stopped. And it was basically the visualization. There were no stats, no theory of germ, it was the visualization that did it, that enabled him to discover this. And in fact, that then led to things like the germ theory and so forth. Another example, dear to many of our hearts, you've probably seen this if you're as obsessive about watching COVID as I, but there was once vaccinations started, there's a lot of question of how effective are they? And back in December, I think December, September, sometime late last year, there's this interesting finding that came out of Israel, which a bunch of anti-vaxxers leapt on, which is basically pointed out that, look, in Israel, there are more people who were vaccinated who have severe COVID than who are unvaccinated. And this is real data. They're not making up this data. There's what's going on. Well, if we, again, let's stay away from stats. Let's visualize different aspects of this data. And you start to see that there's something weird about this when you break it down. So if you break it down by age, you can see at every single age bracket, in fact, there's many more people who aren't vaccinated who get severe COVID than who are vaccinated. The aren't vaccinated are the black, and the green is the ones who are vaccinated. So how can both of these be true? Well, it turns out that the issue is because that first graph looks at absolute numbers, the second one looks at percentages. And when you realize that two things are going on. First of all, in Israel, the vaccination rate was super high. So most people were vaccinated. And also the oldest people were more likely to be vaccinated. And when you have that interaction of factors, then basically it means, of the vaccinated people, there's such a high percentage of population, even though, more of the people who had severe COVID, even though if you were vaccinated, you were less likely to have severe COVID, there were many more people who were vaccinated. So that's why you got that. So again, the point is that you only see this by actually doing that visualization and breaking it down. And I'm going to argue that a common workflow in science, and I mean, this is what I was taught and that I think many do, actually does not support this, right? So a common workflow is something like this. You do your experiment. If you're good, you pre-register it and your analyses and so forth. Then you run your analyses, you write your paper, and while you're writing your paper, or maybe even at the end of writing your paper, you make your figures, and your figures are designed to sort of basically fit with the paper and the story you've constructed. And I would argue that is totally not what you want to be doing. You start with the experiment, as always. Can't get around that part. But I would say the next thing, before you do any statistics, before you do anything, is you make figures. And in fact, you play a lot with figures. So I actually spend about 80% of my data analysis time doing the figures, not doing any stats. And I'm a stats professor. So, I like stats. It's not because I don't like stats. It's because the figures are much more informative, right? And only then do you do the stats. And then you write your paper, and that's when you fine tune your figures to make them look nice. But you really need to have that play figure element there. So what I wanted to do with the rest of my talk, given time constraints, obviously I can't actually give you the technical skills, but I want to give you just a little overview of the kinds of things you can do and give a few tips. So on the technical end, you pretty much want a tool that is, combines ease and power, and these are trade-offs to some extent. I'll be talking about R because that's what I use, but I think, MATLAB has a lot of these same skills, same tools too. And again, I'm mainly going to point you to resources rather than go through details of R. So, but I want to illustrate the sort of kinds of thing you can do with sort of some fake data. My kid is way into Plants vs. Zombies. So, let's imagine that zombies are taking over the world. We've got data at great peril to ourselves. So we want to make sure we analyze it correctly, right? And this is our data. If there's a way to make that available and you want to play, you can, but I just made up this data, right? And we just basically, every year we captured a zombie. There's different kinds of zombies, and we saw how many it killed, and we put little tracking devices on them. Okay. Now, Tidyverse, this package in R, is really nice because you can manipulate data very easily. So for instance, you can take this raw data and very easily say, group it by year, and calculate the mean number of kills per year. I'm including this code again, so you see it, but I'm not going to go in the details. And you can very, very easily sort of say, come up with this summary thing. Okay, that's nice, but we want to actually visualize it. For visualizing in R, there's the most useful package, I think, is called ggplot. And it's kind of a little harder of a learning curve, but the power makes up for it. And it's not actually that hard. It's basically a grammar of graphics. So that means that essentially it thinks of graphics as layers, and the individual things you can do can add on to the layers, just much the same way as when you're painting. you start, a good painting is actually made by layering it rather than putting everything on at once. So, so let me just walk through making a figure in ggplot. So, first the function itself doesn't do anything. It just says, I'm going to make something, give it some data. Again, doesn't do anything. It needs to know what to do with it. The first thing you want to do with it is basically tell it, what are you graphing? In this case, maybe we want to look over how do kills change by year? Are the zombies getting better or worse at killing us? Then what the key thing about ggplot is it has a whole bunch of set of what are called geoms, which basically just tell you how to present it. So this is a column geom. It basically presents bars. So now it shows the bars. This is really ugly, but we can see, hey, not much change over a year. Then you can add aesthetics to your geom, which basically makes it different colors, it affects what it looks like in lots of different ways, depending on your geom. You can have additional geoms you layer on top. So in this case, I've put an error bar on top. There's themes that make it look nicer or not. And you can add labels and so forth. So you can basically build up plots in this way. Again, I've pointed to resources for if you want to learn how to do this, but what I kind of want to end with is just some tips for making these good figures, kind of active, critical, aesthetic tips. And the main thing is that what figures you make should be guided by your scientific questions. So it's really an active back and forth. But one of the main things that I don't see people doing enough is that they're not breaking their data down enough. It's all just summaries. So, in my example, I summarized by year, but maybe, if we look by, each different kind of zombie, we would see something different. So again, that's very easy to do in R, just group by different things. In this case now, I've got the same plot, but I just added a little fill. So the color is, different zombies are different colors. That's a little hard to see. So another thing that R lets us do very easily that people should do more is basically put things in different facets. And now it really jumps out at us. You can see our gargantuars are getting much better at killing us. The normal zombies are getting much worse, and the imps are kind of crap all the time, right? But the point is that was completely hidden before we actually did the proper visualization of breaking it down. Another thing that people don't do enough is that they basically only show summary stats, and we really want to show distributions as much as possible. So here I've just overlaid the actual data points. And you can see just by looking at that basically the normal zombies are, much tighter. So they have a much smaller standard deviation. That was kind of visible in the error bars before, but it's not, it's not so much as easily now. Another tip, sorry, how much time do I have? I can't see the chat.
3 minutes.
3 minutes, okay, good. So another tip, and this is kind of on the breaking down theme that people do not do enough, is that you should really look at individuals as much as possible, even if you have an experiment with like 100 individuals. And I'm demonstrating this with one of my own experiments. I didn't want to make a data set with 100 zombies in it. So this is an aggregate experiment where I was just looking at sort of, you can think of it as a generalization curve. So this is the aggregate thing. Now, one way to break it down is to look at sort of, here's the same aggregate, but I'm posting all the individual data points. That shows a little bit. So you can see basically that, you've got this aggregate, but there's quite a range, especially on the high end. But looking at the actual individuals really points out an entirely different story. And this was the story of our paper, which is that, there's actually a wide variety of distinct patterns that people do. And very, very often, I think we miss that story and tell an aggregate story that's actually not what any single person is doing or single trial is happening. And so you really want to look at those things. The other thing that I want to say is if you need ideas, for exploration, the kinds of things you can look at are very much licensed by different tools, and R has tons of packages. And so I'm just going to sort of point out some of them to get your appetites whetted. But again, have a look at these resources, which sort of walk you through it more. So some of the geoms that R has that are really nice, in addition to the ones I've shown, is like violin plots, which show distributions, box plots. And in this case, this is a great figure, which basically plots them on top of each other. So you see the shape, you also see the median, you also see the mean. Those are really nice. Another thing that R has kind of natively is it will naturally do interaction plots. So you have an interaction. It's really easy to sort of show that and also show all the individual participants too. I love these things. These are so pretty. Rain cloud plots. They're a really great way if you have distributions. It sort of basically shows, this is all the same data, but this really shows the overlap and nature of different groups. Another nice thing, I just like these are pretty ridge plots. If you've got a whole lot of distributions, it'll, all at once, it sort of nicely makes these. Again, all of this is ggplot. These are just different geoms. They're really easy to make once you've got the basic idea. These I've never had cause to use, but I've always wanted to. Like if you're looking at time series data of change over time, a really nice way of visualizing that is sort of looking at changes of groups using what's called alluvial plots. So that's another thing. A few more things and then I'll end here. Again, I just want to throw out ideas. Another thing I see people do all the time is they show correlation matrices as tables. My God, please don't, right? I mean, show it as a picture. It's so much easier to see patterns if you do things like that. And there's a really nice package again in R that lets you do that called core plot. There's actually several different ones for showing different things. And then finally, there's also packages in R for even doing the pure stats. So this is sort of, you run a linear regression, there's a bunch of assumptions you need to check. So this is a package that basically will check all your assumptions for you and make really nice figures of how of it. So, like at the push of a button, you can see all of those things. And again, it sort of visually just jumps out at you. Has it violated this or not? So again, sort of knowing, time is up. And I kind of sprinted through things, but I hope that you just end with the basic idea that try to think about data visualization. If you've thought about it as this thing at the end of the process, it really isn't. It is a main tool for scientific discovery, and play with it. This is actually my super funnest part as a scientist, just playing with your data. And that's why I have all these pictures of kids playing, because that's really what it is and what science is. So I will stop there.
Thanks for that talk, Andy. That was a really beautiful talk. I really enjoyed it. We've got a couple of minutes for questions. I think there was one in the chat a little while ago. I saw that was the first one to come up from Hannah to Aron. Could I ask if you've tried IDEs like VS Code to cut down the toggling between RStudio and MATLAB?
Good question. No, I haven't. So Visual Studio, I haven't tried to use it. I don't think I've had any need to. I think it would have probably overcomplicated things for me. I have heard it can be tricky to implement, but I haven't done it myself. So not sure if anyone else out there has done it successfully.
Not personal, but the next question comes from Johanna Bayer. Thanks for all these great talks. I have a question for Aron again. Any tips on the grade of modularity of code? Should I have one script per analysis pipeline, or would it be better to have several code snippets or functions that can flexibly be excluded after each other?
Yeah, good question as well. I think it's hard to say, and I think a lot of it's personal preference. I see people do it both ways. For me personally, I like to have sort of one script that does a set of very related things. So sometimes that means having multiple scripts, which can be a pain, but then I know that each of those scripts performs very similar tasks. However, I know other people like to join them all together. For me, that's what works best. But I think then it's just a matter of making sure that you keep track of your scripts and label them sequentially so that you know which ones to do and in what order.
I have a thing to add there, which is, I agree on the personal preference. I found it quite useful conceptually to distinguish. There's steps for scripts for cleaning the data, scripts for putting, and that's just sort of, taking out all the cruft and everything. Steps for putting things in the form you need for the analyses, and steps for doing the figures. And I actually put those, I usually have different, R markdown documents for that, or scripts for that. And that's kind of a distinction I find useful, because then I can, clean the data, and that's usually a very separate thing than the others. So, yeah.
I might jump in there, Andrew, and ask a question. I'll combine two at once. So what's your process like for figuring out how best to design your figures and represent your data? Do you have a set process where you want to do certain things first and then you work through it?
Yeah, it's hard to say this generically because so much is about what question you're asking. But usually what I do is I start with trying to look at the aggregate that hits my key questions. And then I think about ways I can break down. So like say the main conditions, the main means, and then I say, okay, what are individuals doing? Is that changing over trials? Is that sort of, and then I also think about different variables that might not be obviously in there, but like, hey, I'm just looking at this thing, but what about the difference between these things? That may be that, right? So yeah, stuff like that. But it really is kind of, yeah, exploration.
I've got a question for Angie, if that's okay. So Neurodesk seems really well suited for open source software and software that's freely available. But what about commercial software? Like for those of us who still use MATLAB or Brain Voyager, for instance, and require an individual license, how can that be used on an open platform?
Yeah, so we have got, there's a couple different options that you can use for a lot of sort of proprietary, well, a lot of software that runs in MATLAB, like EEG Lab, Brainstorm, Field Trip. We actually have compiled versions of those, so you can either use the GUI interactively inside of Neurodesk, or if you have scripts, you can run those through, I forget what it's called, but MATLAB does have kind of a non-proprietary algorithm that lets you run MATLAB code, but not access the MATLAB interface. So you can run all of those in MATLAB, which is something I often do because I don't work with MATLAB as much anymore. And if I have EEG Lab, for example, files that I need to process that have come from someone else, I like to, rather than installing everything from scratch, I just go into Neurodesk and sort of change the format however I want to and get my files back out again. And it's really simple to do. There is also, I believe, the option to enter in a license key to MATLAB, which is something that some people down in Melbourne are developing, but I think it's not currently available, but potentially soon you'll be able to access the actual MATLAB runtime environment and put in your own license key.
Awesome. I think we're out of time, unfortunately. And have we addressed all the questions?
We have one last one.
Okay. Yeah.
Maybe we could quickly go for it. And thank you so much, Angie. It makes me really much more calm about the fact that when I do a system upgrade, if I used Neurodesk, I wouldn't face the problems I face with MATLAB and code. So our last question is for Andrew from Joseph. Does ggplot2 have good brain visualization tools? For example, plotting EEG topographies and or connectivity matrices overlaid visually on the brain. Do you know?
I'm not 100% sure because I don't do them, but I believe so, yes. In that I think I've seen people on Twitter who do neuro stuff, I think, yeah, do. So what I would do in your case is Google R packages and then neuroimaging or whatever, and almost certainly you'll find something because they have basically everything. Yeah.
Fantastic. Thank you. And we know a few people have been wanting copies of the slides. We'll talk to the presenters and communicate with you. We've also recorded this session today, so we can share that link.
Thank you.
Okay, well, that's it. I'd just like to end by thanking Angie, Aron, and Andy, our 3As, for volunteering the A team with their time to share their expertise with us all. And finally, just don't forget to register for the ABPS conference and come to the ECR events. They're going to be lots of fun. Okay, thanks everybody.